Today we are going to start another lecture, that is genetic recombination. Genetic recombination also known as genetic reshuffling. genetic recombination क्या होता है कि reshuffle होते हैं, उसमें reshuffle होना, exchange होना, word for word for the genetic material between the different organisms, which leads to the production of offsprings. It is the process in which different organisms lead to production of the new offspring with their genetic reshuffle or genetic change material in the new traits form that differ from those found in either parents, as from mother and from parents. From father the genes comes, and the new traits which are inherited from parents to offspring are the new traits. So they are genetically recombined or genetically reshuffled traits. In eukaryotes, genetic recombination during eukaryotes can lead to novel set of genetic information that can be passed on from the parents to offspring. From in the eukaryotes, obviously, from parents to offspring, genetic recombination is in the form of novel set, as there lies various chromosomes. Most recombination is naturally occurring, as recombination is naturally by the as chromosomes meet up together. They are in the naturally occurrence. Why in meiotic recombination important? For recombination is important for the formation, first of all, the formation of the haploid cells for the new traits, and for the new copies forms for the new traits origination. These are the reasons that the meiotic recombinations are important. So first of all, in meiosis and mitosis, recombination occurs between similar molecules of the DNA homologous sequences. In meiosis, in the homologous sequences, homologous meaning the same chromosomes lies together, and they recombine and form the new traits for the new version copies for the offsprings. So in meiosis, non-sister homologous chromosomes pair with each other, so that recombination characteristically occur between the non-sister chromatids. In meiosis, as chromosomes are similar, but in the non-sister chromatid form the other chromosomes, they exchange their genetic material. The information transfer may occur without physical exchange. Section of genetic material is copied from one chromosome to another without the donating chromosomes being changed. So physically it is not exchanged like we can see it. We obviously see in the but in the not physically, it's but genetic traits or genetically, or by we can study it's genetically, or by the breaking and rejoining of DNA strands, which forms new molecules of the DNA. So the new copies of the DNA are formed as they are different from the parents. So in both meiotic and meiotic recombination between homologous chromosomes, a common mechanism used called DNA repair. DNA repair, we can called it as genetic material re-exchange, and form new traits with the transfer of materials like genes or alleles or traits are transfer, and so it is also called as a DNA repair. Evolution could not happen without genetic recombination. That's the main point, that the evolution or a change, that sudden change from the after a generation, long time generations, it may change takes place on the large scale, but on the smaller scale, from parents to offspring, simply in a one generation can be seen. If we can see it as eyes color pairs are if both parents have changed, then offspring obviously have like a mother possibility for like a father possibility. But there is possibility lies that different eye color trait originate in the offspring. But there is a proportion as in genetic laws I could explain well. If it were not possible to exchange material between homologous chromosomes, the constant content of every individual chromosome would inevitably fixed in a particular alleles. Okay, now during meiosis in eukaryotes, genetic recombination involves a pairing of homologous chromosomes. This may followed by the information transfer between the chromosomes. Transfer of information between the chromosomes. So, genetic recombination and the homologous chromosomes are in the form of pairing, in the form of always they lie as a pair, but non-sister chromatids will exchange the genetic material as we earlier studied. Homologous chromosomes are chromosomes that are paired during meiosis. This consists of the four chromatids. One pair comes from the father and the other comes from the mother. The homologous chromosomes are the same size. Their centromeres are in the same position, they have the number of genes arranged in the same order. They are usually not identical but carry the same type of information. Each carries the same gene in the same order, but the alleles for each trait may not be the same. For example, two chromosomes may have genes encoding for eye color, but one may have code for the brown color. As I have given you example that trait may change to the next when it is transferred to the next generation, as they may not identical to each other. Both parents have not identical traits, so the trait may change into the next generation as well. As diagrammatically it is obvious that two sister chromatids and the rejection occur, and the sister chromatids take their genetic material, and when genetic material come into the gametes, they are formed four chromatids, and when they recombine, they form the new copies, like with the change of genetic material. So genetic recombination during meiosis, genetic recombination can occur without the breaking and rejoining of the DNA strands. So there is no physical exchange, like one piece of a chromatid or one arm of the chromosome may break and come to the next new and next to the neighboring one and change the genetic material, change the arm of the chromosome, but change of the exchange of the only genetic material genetically. Okay. And when non-sister chromatids intertwine segments, the chromatids with a similar sequence may break apart and exchange in a process called recombination or a crossing over. Like a crossing over may occur. It may not be called as its genetic change of exchange of arm, but it is the change exchange of the genetic material or crossing over the genetic material by the two chromosomes, which may differ, and a new version of a copy formed in the next generation. Okay, now during there is a 1:3 and 3:1 possibility in the offsprings for the new trait. During meiosis, homologous chromosomes exchange a segment through the process called synapsis. Synapsis is the process by which the recombination or exchange of the genetic segment takes place, which allows the pairing of the two homologous chromosomes. First of all, they may lie close to each other and they form pairing and matching up, and chromosomal crossover takes place. That's during totally during a occurring during the prophase 1 in the meiosis. Chiasma is the point where two chromosomes are physically joined. In the first step there is a chiasma take place because there is an exchange of physically joined and genetic material exchange, but not the arm of the chromosome may change. Genetic material will change during chromosomal crossing over during meiosis. Now the next is the homologous chromosomes. These are the chiasma takes place, then the recombination takes place, so the recombinant chromatids are formed. Genetic recombination during mitosis. As we have studied genetic recombination in meiosis, now we are studying about genetic recombination during mitosis. Mitosis is another point. Recombination may also occur during mitosis in eukaryotes, where it originally involves two sister chromosomes formed after chromosomal replication. So in the replication the two sister chromatids are formed and the involved the two, after replication, but in this case, the new combinations of alleles are not produced since sister chromosomes are usually identical. Sister chromatids are identical as we all know about the mitosis is the replicate same copy replication methodology. So there is no no sister chromosomes they are exchange material like this, but they are same copies. Like we have studied that the there is possibility of 3:1 and 3:3 at that time, but in here they are formed like copies. For example, recombination in the organisms with the adaptive immune system is a type of genetic recombination that helps immune cells rapidly diversify to recognize and to adapt new pathogens. Okay, this is some very good example that the adaptive immune system is a good example, as they adopt they know about the fact and figure and know about the disease, so the immune system may develop or new pathogens are also formed by this methodology. Okay, now intermolecular genetic recombination. What do you mean by the intermolecular genetic recombination? Genetic recombination that can occur in different ways. How? Intramolecular genetic recombination may occur in a single crossover or a double crossover. In intramolecular genetic recombination, between the genetic recombination between direct repeats or maybe inverted repeats, as we studied that as we will see in this picture, that the single crossover may take place, so intermolecular single crossover. And now there is in a B there is a double crossover. Intermolecular they takes place in the direct only when one tandem repeats, they may exchange, they may form new recombinant, and in an inverted they may invertly repeats, they may exchange the genetic material. Okay, in the causes of the genetic recombination, what are the causes? These are the causes like crossing over may take place, genetic conversion may take place, genetic reformation can transformation can conjugation, like two individuals are like close together like a paramecium, they have only one cell and joint exchange, like when they combine, that's the exchange of material is called genetic conjugation. Transduction may also occur.